So we are most grateful Dr. Martino is presenting this evening. He is an associate professor in the Department of Clinical Neurosciences at the University of Calgary and a full member of the Hotchkiss Brain Institute. He is also director of the Movement Disorders Program at the Department of Clinical Neurosciences, again at the University of Calgary. And I hand it to you, Dr. Martino.
Thank you, Julian. Good evening. Let me share the screen. Okay, and just gonna. There we are. Okay, can you see the slide? Okay.
Yes, we can.
Yes. Okay. All right. So, all right. Let me know if there's any problem in the process. So
I will do.
Thank you very much. Good evening, and I'm really pleased to for this opportunity to chat with you about a topic that it's quite modern in a way, which is the use of non-invasive brain stimulation, primarily for the treatment. At least this is the final goal of introducing these technologies as therapeutic options in Tourette and obsessive compulsive disorder. And of course, the questions are, why is it relevant, and how are we investigating it in our province? So before getting into the gist of what these technologies are, I really wanted to give you just a preamble of what the basic physics phenomena are at the origin of what we mean as non-invasive brain stimulation. And I promise I won't get into deep into these aspects, which, by the way, not being a physicist, I don't own too much, but this is really a historical preamble that really should begin with this gentleman here in 1831, universally known British physicist Michael Faraday, who discovered electromagnetic induction, in which a magnetic field that varies its position in space can induce an electrical current in a conductor when this magnetic field is placed within the field, when the electric conductor is placed within the magnetic field. So this gentleman really developed this law, which is called the Faraday's law of induction, whereby electric current is in a voltage, basically, is induced in a circuit every time there is a movement, a relative movement between an electric conductor and a magnetic field. And the magnitude of this voltage, of this electric current, is proportional to the rate of change of the flux, basically the rate of movement of the magnetic field in relation to the electrical conductor. So this basic physical property is especially important for its application to the brain tissue, because the brain makes constant use of electricity to convey information rapidly along the very long terminals of the main brain cells, which are the neurons, and these terminals are called axons, and along them travel electrical current travels in the form of what we call action potentials. So we can say that these axons and the sheets of myelin that enfolds the vast majority of them inside the nervous system are really elegant biological examples of very efficient electrical conductors. So we can apply electrical stimulation or magnetic stimulation, and by these means we can alter the activity of these cells. However, it may not come as a surprise that applying electricity or magnetic signals to heal body tissues where electricity is propagated, especially the human brain, is really not a recent accomplishment of medicine. Not at all. I mean, humans have been attempting to use electricity to heal the brain since long before the discovery of electricity and itself, and the basic laws of electricity, let alone of electromagnetism. So, for example, you see stone carvings from the 5th dynasty of Egypt that depict an electric fish that's used to treat pain. And at the time of Socrates in the ancient Greece, in the 5th century BC, electric fish were also used to treat headaches and arthritis. So for those of you who are familiar with cinema, there is clearly it's time for a big time jump. So from the electric fish, this is where we are now. The non-invasive brain stimulation technologies are pretty much this series of techniques that have the potential to modulate brain activity, particularly modulate the plasticity of the brain that the brain utilizes when learning new skills, new motor skills, new cognitive skills. And they do this in a painless way, which means that they don't cause damage to pretty much any tissue that it's between the machine and the brain. So they don't cause damage to the skin, they don't cause damage to the bone or to the brain itself. So they really consist in applying either low intensity magnetic fields, and really low intensity is kind of the buzzword here because this is why they are non-invasive, because they use low intensity of magnetic fields or electrical currents. So we have low intensity magnetic fields used in transcranial magnetic stimulation. We have low intensity electric current used in transcranial current stimulation. And more recently, and hopefully more in the near future, we can even use focused beams of ultrasound to stimulate specific regions in the brain. So let's start just by looking at what these techniques are, briefly how they work. And we start from transcranial magnetic stimulation that we'll call from now on TMS. Now, TMS is really a procedure that is non-invasive and as I said, uses magnetic fields to stimulate nerve cells in the brain in order to change behavior or to improve symptoms of pathological or conditions of disease. Currently, it is approved fundamentally for the treatment of depression, but is also heavily investigated in conjunction with rehabilitation in the context of many other neurological conditions like stroke, brain injury, or other brain lesions. And typically we resort to TMS when other treatments haven't really been effective because it's quite new and to some degree still experimental. Now, an important thing about, so we use TMS to study, so neuroscientists use TMS also to study how the brain works, so to study the physiology of the brain. However, when we investigate TMS as an intervention, as a treatment, we use it in a very specific way whereby we deliver repetitive pulses, repetitive magnetic pulses. So we can say that the treatment form of TMS is called repetitive TMS or rTMS. So what happens, as you can see in the picture, there is a coil, an electromagnetic coil that contains the magnetic field and delivers it. It's placed against the scalp. As you can see, there's really nothing between the scalp and the coil. And it's placed at different possible regions, and where we position it depends on which area in the brain we want to target, and this depends on which condition or which manifestation we want to treat. So this electromagnet delivers in a painless fashion a magnetic pulse that stimulates nerve cells in a precise region, precisely in our target region. And because we need to do this over and over again in different sessions, this is done consistently. It's happening consistently in the same region. And we can actually achieve this by using MRI images of the brain that help us navigate the coil in the right spot, so that we consistently stimulate the same region over and over again across the different sessions. This is definitely a constantly evolving technology. There are different ways to perform this procedure, and techniques may change as experts learn more about the most effective ways to perform these treatments. Now, this is a busy slide, but just don't get discouraged. I'm just going to focus on giving you some general further input on how TMS works, which is not still completely well understood, but to some degree it is. And the basics information is what I would like to highlight now, which is, as we said, the TMS is based on the application of a magnetic field. There's a coil that is placed on the participant's head, and this induces an electric field in the brain, again, via the Faraday's law that I mentioned at the beginning. So this induced electric field may trigger action potential, but the way we use it, it's meant to alter somehow how excitable that region is. So alter the neural activity of our target region. And the effects of TMS depend on many different factors. They depend on where we stimulate, so which target region, how we position the coil, with which angle on the scalp, the intensity of the stimulation that we use, the frequency of the pulses that we deliver, and the overall number of pulses that we deliver. So repetitive TMS, we said, is the therapeutic form of TMS, and it can either increase the activity of a brain region or suppress it or inhibit it. Because there are situations where for treatment purposes, we want to excite a region that is underperforming or suppress a region that is overactive. And the way we actually proceed with this is based on the frequency of the pulses that we deliver. So we have when we use so-called high frequency repetitive TMS, which means 5 hertz or above, 5 pulses per second or above, we tend to obtain an excitatory effect. Whereas when we use low frequency, such as 1 pulse per second, 1 hertz, we tend to obtain an inhibition of this region, of the target region. There are also more complex ways to deliver our TMS that use very high frequency, like 50 hertz, but for a very short period of time with very short bursts. We call this theta burst, and this seems to be one of the forms of the TMS of repetitive TMS that produces the longer plasticity effects over time. What about transcranial current stimulation? So we're talking about transcranial magnetic stimulation. Now, transcranial current stimulation does not deliver magnetic fields. It delivers so electric currents. And the most used form of transcranial current stimulation is the so-called TDCS, so transcranial direct current stimulation, whereby, again, it's a non-invasive stimulation that it's, again, painless and uses direct electrical currents to stimulate specific parts of the brain. And the current, again, is very low intensity. It's between 1 and 2 milliamp usually. And it passes through 2 electrodes that we place over the person's head. One electrode, which is the active electrode, and the other electrode, and the active electrode is positioned on the same region that we target for TMS, whichever that is. So that's going to be our target region where the coil in TMS is placed. We position this active electrode, and the return electrode is on a bony region that is not in conjunction with any brain part. It's either forehead or behind the ear on the mastoid. And there are two types of stimulation that we can use with TDCS. One is called anodal, and one is called cathodal. And this is determined by which electrode is positioned on the target region. So we call anode the positively charged electrode, the one through which the current enters a polarized electrical device and a circuit, whereas the cathode is the negatively charged electrode through which the conventional current exits the electrical circuit. So so we have when we apply anodal current stimulation, it means that we put the anode on our target region, and this is the form of TDCS that increases the excitability of that region. Whereas when we position the cathode, we decrease the excitability of that region. And as you can see, really, this is the circuit. There is a stimulator that sends the constant current into the circuit, and the circuit is closed between the two electrodes by the brain tissue, obviously, because there is a flow of current through the head that targets the brain tissue that, in the areas that we wish to target. So this is definitely a more experimental form of brain stimulation than TMS, but it has a number of advantages, potentially. It is cheaper. It's easier to administer. As you can see, the equipment is pretty small and it's easily portable. It can be exported at home. Works a little bit like a TENS machine that sometimes you might have heard to treat nerve injury or other form of sports-related injuries. And the most common side effect of TDCS is really only a slight tingling or itching in the region that is stimulated in the scalp, which typically goes away a few minutes after the end of the stimulation. But unlike TMS, this is not yet approved for treatment. It's still an experimental therapy. Now, I mentioned at the beginning that we have three different forms of stimulation, and the third one is really the futuristic one, the one that we are not doing yet. And this is what we call the low intensity, low frequency, focused ultrasound stimulation, or LILFUS. Again, so this is a very complex device through which beams of ultrasound are conducted throughout the skull, throughout the head and the brain, and they can reach, unlike TMS or transcranial current stimulation, they can reach pretty much, in theory, every region in the brain, including very deep structures that the TDCS or the TMS cannot reach because they are too deep and far from the skull. Now, there is some initial evidence that by applying this low intensity focused ultrasound, we can present, for example, to the sensory cortex or to another deeper region called the thalamus, we can change the sensory discrimination abilities of an individual. We can decrease pain. There's very initial evidence of that, but this is still not applied, if not in very, very limited experimental studies. But the focused ultrasound program in Calgary has developed a method that improves the ability to convey these ultrasound beams precisely to the focused region of interest. And hopefully we will exploit this new technology in the next couple of years. So I think allow me a little digression before we move on to talk about how these techniques have been applied so far to Tourette or OCD. Just to clarify that you will hear a lot about neurostimulation and non-invasive brain stimulation, not only to treat conditions, which means to restore a normal functioning in people who suffer from symptoms from neurological or psychiatric or substance use disorders, but also to enhance the activity of the function of individuals who are not affected by any of these conditions. For example, to improve cognitive skills, to improve motor skills, to improve memory, to improve the ability to control one's emotions. Now, these enhancing approaches are very popular and have also led to the promotion or the marketing or the commercialization even of do-it-yourself devices that do not have a very solid evidence basis. So this, on top of that, there is also this has also led to a number of ethical discussions about how appropriate it is to apply these technologies to change normal aspects of emotion and behavior, like, for example, to enhance decision-making abilities, to reduce fear, to improve cognitive motor skills. So this is a quote from a scientific journalist covered by a piece on the New York Times some time ago, that says, what role does doubt and fear play in our lives if its eradication by neurostimulation techniques actually causes so many improvements? Do we make more ethical decisions when we listen to our inner voices of self-doubt, or when we're freed from them? If we all wore these caps, would the world be a better place? And again, is brain boosting a fair addition to the cognitive enhancement arms race? Will it create a social divide where the rich can afford to be smarter and leave everyone else behind? Will tiger moms force their lazy kids to strap on a Zappity helmet during piano practice? I mean, there's a lot of discussion of the potential growing uses of these techniques, but the take-home message is that these are really technologies that should be tested in rigid and rigorous experimental contexts, and that they really should be assessed by the public, possibly through the filter of neuroscientists who know how to handle these technologies and interpret what these technologies can do. So this is a small digression that I felt it was important to make at this point. But then let's move on to see what are the potential therapeutic applications of non-invasive brain stimulation to Tourette and OCD. And I used before at the beginning the term plasticity or neuroplasticity. And this is really crucial because it really points to the way that we should be using non-invasive brain stimulation as a treatment, as a potential therapeutic tool, which is to modulate the activity of the brain while an individual is already engaging in a learning process, is already learning or relearning a motor skill, a certain cognitive task or a set of tasks. Or more relevant to Tourette or OCD, for example, is undergoing behavioral therapy, and it's learning how to control tics or compulsive rituals efficiently using a behavioral therapy. So plasticity or neuroplasticity is the ability of brain networks to change through growth and through reorganization. And these changes can happen from individual nerve cells or neurons that form new connections with other neurons, but it may also be that the whole networks in the brain can undergo a reorganization, remapping. So examples of neuroplasticity include changes that result from learning a new skill or learning or practicing, but also through environmental influences, medications, psychological stress. And neuroplasticity originally was thought to by neuroscientists to manifest only during childhood, but we know now we have learned over the past decades that many aspects of the brain can be altered are in a plastic way even through adulthood. However, we should always keep in mind that the developing brain really exhibits a higher degree of plasticity than the adult brain. So the general concept is that that non-invasive brain stimulation technologies work better and have a better and beneficial impact on improving plasticity when they are coupled to specific learning processes, like, for example, in the case of tics and OCD, again, going through a behavioral treatment. So how has this been tested or investigated so far in the literature, in OCD and in Tourette syndrome? So we have a specific set of brain regions that in OCD or in Tourette we can target with these technologies, and these are the three regions that have mostly been tested in OCD, and to some degree some of them also in Tourette. And these are this first region, which is a motor region called supplementary motor area, or SMA. This is a region of the brain sitting right in the midline of the brain, and it's important for motor control. It controls movements that are internally generated rather than directly triggered by sensory experiences. It also coordinates movements that are performed by both sides of the body, like movements that involve both hands or sequential movements. Then we have this other region, which is called DLPFC. It's the most anterior region. It's also known as dorsolateral prefrontal cortex, which is involved in a number of executive functions, like working memory, like shifting from one task to another, planning things, suppressing unwanted behaviors or movements, abstract reasoning. And it's also involved in higher level of motor planning and organization. And this is also the target for TMS in depression. And finally, we have this emotional region, or orbitofrontal cortex, which is an area of the brain that is really sitting just above the ceiling of our orbits. That's why it's called orbitofrontal. And it's also involved in modulating behavior when this is related to seeking a reward. And it's involved also in decision making. Now, all these different regions have been tested with rTMS in OCD, and pooling together the data that we have available, there seems to be a benefit of suppressing this region, this motor region here, or the orbitofrontal cortex, or exciting the dorsolateral prefrontal cortex using rTMS. However, of all these regions, the one that has reported most consistently beneficial effects in OCD is the orbitofrontal cortex, which seems to help more reducing symptoms. Oops. So the same regions have also been targeted with TDCS in OCD, but the level of evidence that we have in applying transcranial direct current stimulation in this condition is lower. We really have case reports or case series. We don't have randomized trials, which represent a higher level of quality of evidence. And again, these preliminary results coming from this case series and case reports really show that if we target this region, the motor region here, or the orbitofrontal cortex, we can produce significant benefits to obsessive compulsive symptoms. But there is no doubt that these studies have published case series of patients that were very different, different numbers, different ways to perform these treatments. So different duration of stimulation, different number of sessions, different intensity of current. So what we know about the application of this technology is promising, but it's certainly not enough. And most importantly, none of these studies has coupled either of these two techniques, the TMS or the TDCS, with behavioral therapy, which is a major limitation of what we know so far. Now, since we're talking about OCD, I just wanted to also make a little clarification, because you might have heard that here in Calgary, there is also an ongoing study that is using a different type of stimulation, which is also a focused ultrasound stimulation. But this time, it's not a low intensity focused ultrasound stimulation. It's a high intensity focused ultrasound stimulation called HIFU. Again, it's done at the focused ultrasound program in Calgary, but this is an invasive procedure. It's surgery. It's not a non-invasive procedure, because the intensity of the ultrasound that is delivered is very high. So we generate a lesion in the brain, in a specific area deep in the brain, in order to treat severe forms of OCD, which is resistant to medications or to behavioral therapies. And adults are being studied with this treatment. If they have, if they are qualified, if they're eligible, if they have refractory obsessive compulsive disorder. And you can see here some of my colleagues who are performing this study, our neurosurgeon, Dr. Kish, and Dr. Pike, who is the director of the focused ultrasound program. But again, bear in mind, this is not non-invasive brain stimulation. This is invasive, because we generate a lesion in the brain by doing this. So just a brief mention to say that these technologies have been tested in ADHD as well, because ADHD obviously is quite common in Tourette and OCD, so it's worth mentioning. However, again, there's no clear evidence of efficacy of RTMS in ADHD. There's no clear evidence that when we deliver real TMS compared to sham stimulation, we achieve a benefit in ADHD. So we need more investigations. The number of studies is actually very limited, and therefore the literature is not conclusive. There's perhaps a bit more that has been done using TDCS in ADHD. There are some studies that have shown a partial improvement of hyperactivity and some cognitive deficits that are related to ADHD, particularly when stimulating, interestingly, with the dorsolateral prefrontal cortex, which is the target of depression too, which is interesting. But and again, the type of stimulation is excitatory. It's anodal TDCS. But its routine clinical application in ADHD is definitely not recommended. We definitely need more research to learn which are the optimal parameters of stimulation to improve the efficacy on clinical symptoms. What about tics? So again, most of the application that has been published so far of these technologies in tics and Tourette are really based on TMS primarily. And so our repetitive TMS as targeted fundamentally with the greatest level of promise, the supplementary motor area, that motor region that sits in the midline of the brain, in the internal or medial part of each brain hemisphere. And this is the hot target area for tics. And this is also because brain imaging has demonstrated that the activity of this region changes in association with the release of tics, as well as also with the inhibition and suppression of tics. But the general agreement that we should try to suppress the activity or the excitability of this region in order to try and reduce and diminish the severity of tics. However, the studies so far have not been clearly conclusive. They have particularly shown that if we stimulate with active stimulation for long enough, for several weeks, we can observe a decrease in tics. But this is particularly observed when people with tics are know whether they are know that they are receiving active stimulation. So when we are performing a comparison between active and sham stimulation during this blinded period, so far we haven't, these studies haven't really demonstrated a clear benefit with the real stimulation compared to sham. But the studies so far are limited methodologically, and again, they were not conducted coupling this stimulation with the behavioral therapy, which is a major point. This is another interesting approach, which is not exactly neurostimulation, but just to show you that there is another technique that's called neurofeedback that is really in its dawning age, which really uses MRI, uses functional MRI signal to teach subjects to control the level of activity of target brain areas. So these are studies that have been conducted in recent years in Yale. The idea is that patients are given a stimulus to which they have to respond in order to reduce the level of activation of the target area in the brain. The SMA, in the case of tics, the supplementary motor area, or the orbitofrontal cortex in the case of OCD. And they can actually follow the level of activity while they are in an MRI scanner. And they receive, in these studies, they have received a couple of sessions within 1 week. And what was really surprising and interesting is that the application of this neurofeedback technology led to a reduction of tics and obsessive compulsive symptoms severity. But this reduction in severity, interestingly, continued to occur even weeks after the treatment week, the week where the treatment was delivered. So this was really, these studies really need more control, more to be confirmed and verified by new studies. But this approach looks very interesting and promising. Also, it's also highly non-invasive, and it's a form of neurostimulation. Well, it's a form of neuromodulation that I think it's worth mentioning. So what are the studies that are happening in Calgary with respect to non-invasive brain stimulation? Particularly, there is ongoing research on Tourette syndrome and tic disorders, which, as we know, are network disorders. So when we talk about both Tourette syndrome and OCD, we know that a whole network is affected. And but within this network, the supplementary motor area, again, appears to be a crucial region for the modulation of tics, or for the modulation of urges to tic and the ability to suppress tics. So as I said before, so far, these non-invasive brain stimulation technologies have never been performed in conjunction with behavioral strategies. And this is what actually one group is currently working on. This is Dr. Frank McMaster's team, who is conducting a study which is funded by the Canadian Institutes of Health Research in order to apply repetitive transcranial magnetic stimulation in conjunction with the comprehensive behavioral intervention for tics, the CBIT, which is the most used behavioral therapy intervention for tic disorders. And they are recruiting children with Tourette syndrome between the ages of 6 and 18 who haven't had more than 4 CBIT sessions prior to enrollment. And the treatment involves a combination of 8 hours of CBIT therapy and 20 hours of repetitive TMS therapy over the supplementary motor area. The study involves, so the CBIT therapy is delivered over 10 weeks and the TMS therapy is delivered over 5 weeks. Sessions last an hour each, and at the same time there's a lot of data collection from participants that include mental health assessments, brain imaging scans, brain activity assessment. These are the contacts of this research team. Obviously, I can definitely, I know that this information is already circulating, and this is all leaflet material that the team has kindly shared with me for this presentation. But obviously, if you're interested, I believe that you can contact Julian, as well as also Dr. Pringsheim, who is a co-investigator in this study. I am personally involved in a smaller project, which is still at a more initial stage compared to Dr. McMaster's. That is instead applying TDCS, so the transcranial direct current stimulation form of neurostimulation. Again, just like for RTMS, we are looking into an inhibitory or suppressive stimulation of this supplementary motor area, so cathodal TDCS of the SMA. And there is initial evidence coming from an English group in Nottingham that has shown that applying a single session of low intensity cathodal TDCS over this region can acutely decrease the severity of tics for a period of 90 minutes. This is the effect of a single session. However, really these technologies it makes sense to use them when we deliver repeated sessions, because the effects can increase session after session. It's what we call the cumulative dosing of brain stimulation. On top of that, we know that a technology, an intervention like this, is more efficient on the target area or the target network when this network or this area of the brain is already engaged in some form of activity. And this is the principle through which we have to engage in cognitive or behavioral tasks the target region when we apply non-invasive brain stimulation. And the idea is indeed, like for RTMS, is to couple TDCS with the delivery of competing motor response training, which is obviously the main component of habit reversal therapy. Participants to this study are provided some structured training on how to develop strategies to counteract their tics through the use of competing motor responses for at least 6 to 8 weeks. And then they are asked to implement these motor responses, to use them, to apply them during the stimulation sessions. And the stimulation sessions are provided over 5 consecutive days to participants with Tourette syndrome who are 16 years of age or above, so older adolescents or adults with stable treatment. But this study is a randomized study. It means that participants are randomized to receive either the active stimulation or a sham stimulation. And some of them get also a brain scan to see if the real stimulation can modify the connectivity of the supplementary motor region that we are stimulating. So this is just a very, very preliminary and therefore not conclusive snapshot of the first 12 patients pretty much 50% of the number of people we would like to recruit eventually in this study, which is also conducted in collaboration with Dr. Pringsheim. As you can see, there is an initial promising signal that the degree of reduction in the severity of tics is greater with a real form of stimulation compared to sham. And this is also, this is interesting because this is the time point immediately after the 5th day of stimulation, and this is the time point, visit 6, 1 week after. So 1 week after the stimulation has ended, you can see that there is the benefit is somewhat sustained, at least in this very preliminary set of data. So this overall leads us to comment that TDCS over the SMA, first of all, has been very well tolerated, at least when administered over just 5 consecutive days. We had transient headache in one participant, and another participant more recently. There has been tingling over the scalp in the region that has been stimulated, which really went away after about 20 minutes. There is an initial promising signal of efficacy in decreasing the severity of tics, but more analysis should be performed on this data, and when the study will be complete, also to see if there's any relationship between the expectation that patients have and the response to the intervention, as well as whether the other conditions associated with tics, the comorbidity profile, in some way influence the responsiveness. But obviously, if this were to be a positive trial, eventually showing a difference between real and sham stimulation, the next step would be to make a larger trial that compares different durations of treatment, and also that might look into applying this technique at the patient's home as opposed to hospital, because this technology, this particular device, the TDCS, can be easily exported, as I mentioned, at home. So this was really my last slide, and this is just this is a very important list of collaborators that have, at least with respect to me, that have worked with me in this throughout this TDCS trial that is currently ongoing here in Calgary.